Good afternoon, everybody. It is Wednesday, the 24th of February. As advertised, today we have with us Dr. Richard Millis. He's been with us before. He's a researcher in the world of medical physiology. You've seen his bio that we've posted for several days, so I'm not going to go into a big, long introduction, but he's going to give us some updates in regard to his work with QEEG. When he's done, he'll let us know, and then we'll go ahead and open up for questions and answers. So, Rick, thank you so much again for being with us. It's always a pleasure to have you, and it's all yours.
Thanks. Welcome, everybody. So the disclaimer, if you showed them what I sent you in terms of my bio, is that picture was taken about 5-6 years ago, and I look quite a bit older since then, even though I'm in the Caribbean, more wrinkles, and so I'm not quite that handsome anymore.
Oh.
Anyway, electroencephalographic correlates of brain adaptations to medical school. And so this is a pilot study. And how it came about is the provost of my medical school, one day, I hope you guys remember, there was a day where we actually worked in our offices. And I was sitting at my desk, and I got this call from the provost, who almost never calls me, so I thought I was in a lot of trouble. And he says, do you know anything about, and he named some guy, and he was actually at the Association for American Medical Colleges, AAMC meeting, and they were talking about brain training to help medical students and speed up learning and all that. He said, do you know anything about it? And I said, well, I know a little bit about it. I've been dabbling in this quantitative EEG with, Dr. Suter, and he's one of the pioneers and gurus in this area. So I know a little bit about it. And he says, well, you might want to look into it because there are people who are starting to look at it. Then I also started looking around at funding possibilities. Even though I'm in the Caribbean, I looked for funding from NIH, Department of Defense, and I found out that there was a broad agency announcement from the Department of Defense on technologies to speed up learning. And I said, well, brain training and neurofeedback is perfect for that. And so I started questioning, would this work for our medical students? And so I designed a little bit of a pilot study. The experimental design was, we found 10 male semester 1 students who just are coming in. They were either American or Canadian citizens, and they're coming to the Caribbean for the first time, generally, at least to go to school. So it's a big adjustment for them. And they're starting medical school. So there's a huge adjustment from undergraduate life to medical school, professional school life. And I thought these would be perfect students to see whether we can see if there are any changes associated with their preparing for their first medical school exams. So we made sure that they had short, little or no scalp hair to just make everything easy, because we were new at this. We only found 10 who volunteered, who were willing to, abide by our supervision for a couple of days when they were being studied by QEEG. So they turned out to be, these 10 students are age 22 to 42, and they all happen to be right-handed, which is significant when you talk about dominance and non-dominance. And then, of course, we'll give New Mind some commercial here at advertising. We bought some Brain Master Discovery hardware and software, which was the first time I had ever used QEEG. And we purchased it through New Mind and New Mind Maps. And it turns out that out of the 10 subjects that we originally recruited, we had 9 that we studied under eyes open conditions and 8 under eyes closed conditions. The only reason we had only 8 under eyes closed conditions is because we found some messed up data that we couldn't use. And so that happens when you do research.
Yeah.
And so eyes open conditions, 9 subjects. So what happened to the other subject? Well, he just dropped out. He just wasn't willing to do almost anything. And so our subjects were studied at 9 a.m. after overnight fasting, no physical exercise. We made sure their body temperature, their vital signs were all within the normal limits. Because I learned a long time ago, when you do human studies, you have to control your controls. You can't control a lot of things, but what you can control, you make sure you control. And if you do that, you might actually get some significant data. We excluded mental or medical health diagnoses, smoking, medications, recreational drug use. We did 10-minute steady state QEEGs. We had the subjects focus on a small black circle drawn on a blank white wall when their eyes were open, and eyes closed was just volitional. We didn't use any kind of covering on their eyes to help them keep it closed. And it was done in a quiet, darkened room, seated in a chair, when their vitals were stable. So we used their vital signs as an indication that they were in a steady state, and we proceeded. Once we got the data, of course, we removed the QEEG artifacts manually. Each subject was studied twice, 1 to 2 days before their first and second medical school block exams that, for the purposes of this presentation, we're going to call B1 and B2 block exams. The block exams consisted of 2 parts. There was a mind, brain, and behavior course and a human structure function course. So we looked at their scores on those exams to see if there were any correlations. And they were 5 weeks apart. At weeks 5 and 10 following matriculation. In other words, following entering school at a Caribbean medical school, away from home, this is when we studied them. In other words, they went to classes for 4 weeks, and then they prepared for this exam, first exam, and then they had a week to prepare, and then another 4 weeks, and they had another week to prepare, and that makes up the 5 and 10 weeks. We also had them do the individual self-inventory of psychosocial interactions from the new mind maps, and they had to complete that within 12 hours of doing the QEEG, 12 hours after the QEEG. They had to complete it, and we averaged their 2 inventories, at least the scores for their 2 inventories. Now, you might say, well, how did you get scores from the inventories? Because they're only graphically represented. Well, we use the graphical representation to extrapolate the scores. We think we did a pretty good job of that, but who knows? Let's see if we did. I'm going to share some raw data just to show you how we go about doing research. Here's our eyes open condition with our 9 subjects, and here's our eyes closed conditions with our 8 subjects. And notice that for the mean delta amplitudes, this is the blue is the block 1 exam. That is 1 to 2 days before the block 1 exam, and the brown is 1 to 2 days before the block 2 exam. Remember, we had those 2 exams. So we're looking at the time for preparation between these 2 exams. Okay? So this reflects the difference between the block 1 score and the block 2 score for amplitude, delta amplitude. So each subject, some went up, some went down, some went up, some went down. Nothing significant here. Let's look at theta. Wow. Theta looks better, doesn't it, in terms of a pattern? In terms of a pattern, they're almost all going up. Eyes open and eyes closed between block 1 and block 2. Let's look at alpha. Well, alpha looks pretty good too. Okay, but then we have some of these that are not very consistent, messed it up, so we really didn't get anything significant out of alpha. Let's look at beta. Let's compare beta to theta. Well, it's interesting. Here we see a nice pattern, and in beta we see a nice pattern too, don't we? So that's what we found. We got significance out of theta and beta. Well, that's convenient because when you look at theta beta ratio, it seems to be a measure of attentional control. Well, can you think of anything better in terms of medical school or any kind of learning than being able to pay attention? One of the reasons why I'm not a real doctor, I'm just a PhD, is I can never pay attention well enough to go through the rigors of a medical school curriculum. So I settled for my PhD, but I'm happy. So we got the theta beta ratio, and let's look at the theta beta ratio. Well, remember what the pattern for theta and beta look like? Looks like we got a pretty good pattern here for theta beta ratio, don't we? In terms of all the subjects pretty much going in the same direction with very few exceptions. Then we wanted to see whether the standard deviation of the theta and the beta amplitudes was going to be a problem, because remember, we're looking at mean voltage here from the FFT analysis from the brain master. And what we do is when we look at that theta, those theta amplitudes, those are, and then we average them. Okay, we average them, let's say, for all recording sites, and that's what we do. And when we average them for all recording sites, we're taking the mean of the mean. So we wanted to see whether that was, there was going to be some variation within that second mean, the mean of the mean, the mean, the original mean had some standard deviation to it, which is reflected here. And we wanted to see whether we'd see any significant pattern, which we didn't, in the standard deviation of the theta amplitudes and the beta amplitudes. And we found nothing, we couldn't find any statistical significance out of that. Then we looked at the coherence. Now we're going to talk a little bit more about coherence later, but coherence is pretty much from BrainMaster correlation coefficient between what's going on in one recording site compared to another recording site, how much they're talking to each other, or what we call, what I think the brain maps system calls interconnectivity. And so this coherence would give us some information, and you see a nice pattern in the coherence, don't you? Very similar to what we saw in the amplitudes. But we'll talk about coherence later, and the beta coherence. So it's amazing, I think, let me take a moment to just digress. I think it's amazing that in only 8 or 9 subjects, when you control your controls, you control the conditions very, very well, all right? And we certainly didn't control age, we controlled gender or sex. We only had males, and we controlled for hair. They had to have short, no, or no hair. But we also, controlled, we didn't control age. So we have a wide range of ages from 20s to 40s. To me, it's kind of amazing that we would see this very, very consistent pattern with just such a small group. Now, remember, we said that the exams were given in 2 parts. One was an MBB, mind, brain, and behavior score. And let's look at their scores, the pattern. This went down, this went up, this went down, this went up, this went up a little, this went down a little, nothing significant. Nothing significant here. But look at their human structure and function exam scores, a little bit more variation. So we looked at correlations between their exam scores, between block 1 and block 2, or both of these, and just by looking at it, I would expect some more significance and some more pattern showing significance from these HSF scores than I would from the MBB scores. But we'll see if that was true. So what did we do? Well, here's the EDF file, the European Data Format file, that of course is standard with brain maps and brain master, and we get it's standard across the world actually for medical related time series data. And so we started with we got the EDF file. From the EDF file, we got the CSV file that comes with the brain mapping. And we started doing some data crunching. So when we talk about amplitude, when we looked at amplitude, we looked at the mean amplitude. We're taking the mean of the mean amplitudes at all the sites. And we just averaged them. We didn't, and at the bottom of each of these CSV files, we developed formulas and we averaged the mean for delta and theta and alpha and beta, and we ignored anything else because those were the standard ones we knew most about, and we expected to see something. And so here we see the mean and the mean standard deviations and the mode frequencies and the coherence. And then we developed some of our own formulas for prefrontal asymmetry and frontal asymmetry and temporal asymmetry and parietal asymmetry and all that kind of stuff. And then we just created an Excel file. So here, this is coherence, delta coherence, and here are our subjects. Here are 9 subjects in the eyes closed conditions. This is block 1 and block 2. I took their names out so you won't be able to identify them. And then eyes closed conditions. As I told you, see this data point is missing. So we really could only do 8 subjects for block 1 and block 2. So here's our Excel spreadsheet we created, delta, theta, alpha, and beta. And so for all of our variables, we had spreadsheets like this. We did t-tests to look for significance. Here's our means. Here's our t-test value in terms of p-values and standard errors. Okay. So then let's remember we're taking this data from the spreadsheet, the BrainMaster spreadsheet, and of course we also created brain maps. And so we wanted to know whether or not the magnitude that we see on the brain maps had any significance in terms of the data reflected in the CSV files that we were analyzing. And so I'm just going to show you one example. Here's one subject, and this is his block 1 magnitude or voltage. Okay, and if we look at the differences between block 1 and block 2 in the eyes closed condition, we see some differences, don't we? We certainly see delta changing. We still see theta changing. Does this theta that's changing between block 1 and block 2 seem to reflect pretty much what we saw in our data? Well, I think it does, even though this is only one subject. Alpha, we see some changes in alpha from block 1 to block 2. And we see some changes in beta. Look, beta going up. So remember, our data was kind of showing a pattern toward theta going up, and theta-beta ratio going down. And that's what we're seeing. Kind of theta going up, not as much, but beta going up a lot. So it makes sense, at least for this subject, that his theta-beta ratio would be decreased. Now remember, decreased theta-beta ratio indicates better attentional control, according to some investigators. So when we did the analysis, and here's block 1, eyes open, block 2, eyes open, block 1, eyes closed, block 2 exam, eyes closed, we see significant increase in the theta amplitudes, just like we eyeballed from the raw data. And the same thing for beta. They're going up. Eyes open, eyes closed, both. And theta-beta ratio. Theta-beta ratio going down. Here's p less than 0.01. To me, it's amazing that if you control your controls in a group of 8 or 9 subjects, even though in 8 or 9 subjects you have very low statistical power. We know there's a type 2 statistical error here, but that's why you do a pilot study, so you can design a better study later. And hopefully you'll get some money to do that, and you'll be able to actually prove anything. Prove something, not prove anything. We don't want to prove just anything. We want to prove a hypothesis. So that's called proving something. Okay. So again, to me, this reflects what we saw when we eyeballed it. So then we said, okay, let's look at the block 1 and block 2 exam scores. Well, interestingly enough, in all the analyses I'm going to show you and we have done, we have seen nothing in terms of significance for any of the block 1, for nothing for the MBB exam. Remember we said the MBB exam, I didn't expect to see anything when we eyeballed it. And so that MBB exam, we saw nothing. For the HSF exam, we saw nothing for block 1. But for block 2, we found something very significant. Now, what could this mean? Well, block 1, remember, is 4 weeks after they're just coming to medical school. They're getting organized in an island environment for the first time, for most of them. They're transitioning from undergraduate to professional school. Now we're throwing a study at them where they have to kind of learn how to sit still and look at a blank wall and have electrodes put on them. So, in my opinion, that's kind of a acclimation. That was an acclimation in the block 1. And it didn't bother me that we didn't see anything significant for block 1, but look for what you see for block 2. So we take the theta beta ratio for block 2, and we compare it to the, or we correlate it with the HSF exam score. What does it show? The worst exam scores, okay? The worst exam scores are here. And those are people with the what? Highest theta beta ratios. And it's interesting that none of these theta beta ratios are enough theta beta to be diagnosed as ADHD or anything. They're all below 2. And so these are all pretty normal people. And yet we see this significant correlation. And we see the same thing whether this is eyes open or eyes closed. This happens to be eyes closed, but we got the same pattern in eyes open with a P level of 0.03. Now we said, okay, let's combine the eyes open and eyes closed data. So when we combine eyes open and eyes closed, we got block 1 theta, block 2 theta, okay, and beta. This is now combined, eyes open and eyes closed, showing the same pattern we eyeballed and we saw before. And the theta beta ratio, the same pattern. Now when we, of course, pool the data for eyes open and eyes closed and do the same kind of correlation between the block 2 exam score and their theta beta ratio, we see the same thing, but much higher significance, greater degrees of freedom, better correlation coefficient, still showing the same thing. What? If you've got a higher theta beta ratio, it seems like you're going to get a much lower exam score. And if that translates to attentional control, which we would like to prove in a future study, give these subjects tests of attentional control, maybe we're onto something here. So, as you know, brain maps gives us an overall change. And again, I showed you that brain map, remember before, from this kid. I shouldn't call him a kid, medical student, showing the change. And this is the same student and same subject, and a 62% overall change, which is pretty large, probably well beyond what you expect from just chance, but it's very interesting. Now we want to see, does that overall change reflected in the theta beta ratio? So we correlated the overall change with the theta change, with the change in the theta beta ratio. Overall change in the QEEG with the change in the theta beta ratio, and what do we see? A tendency, not statistically significant, but we would call this marginally significant, maybe a trend in this direction. Well, this to me is very interesting. I would really like to follow this up with a bigger study because, you know, this tells us the theta beta ratio is kind of consistent. It's not reflected in the overall change in the QEEG, that this kind of stays, you know, about the same all the time, which is good. That might mean it's an important genotypic or phenotypic characteristic that we can rely on, that has some resiliency, some robustness when we study somebody's theta beta ratio in the proper way. Now, the next thing is the interconnectivity, which I hope brain maps use coherence. I took the liberty of saying interconnectivity was a reflection of coherence here.
Mm-hmm.
And again, this is the same subject we saw before. Let's look at their coherence or their interconnectivity according to brain maps, New Mind brain maps. And it looks like that theta is increasing. And connectivity, look, that connectivity is increasing. Let's look at that beta. That beta is increasing. Even alpha. So what does this mean? Does this mean that the interconnectivity, which we think is a reflection mostly of network communication between one recording site or the cortex underneath one recording site compared to another recording site, is this a significant indication that preparing for medical school or going through an academic challenge like medical school, training somebody's brain so that different areas of the brain are talking to each other more? Why not? I like that hypothesis. But then we looked at coherence at specific areas, okay? We looked at the coherence in specific areas. We looked at the prefrontal. In fact, what I'm going to show you now is data that reflects only significant. If you don't see the data here, you don't have to ask me. It's not significant. We did a shotgun approach and we said, hey, let's find anything that's significant and let's put it in. I call this pre-paradigm research. Almost nobody is doing this kind of research the way we're doing it. So it's pre-paradigm research. For a preliminary pilot study, I think it's okay to do a shotgun approach and look for anything that's significant. So that's what we find. One asterisk suggests P is less than 0.05. Here's one that's less than 0.001. 0.05. For our orbitofrontal or prefrontal, orbitofrontal, eyes open. For alpha, beta, coherence, as well as for frontal, F3, F4, beta coherence, where the F3 side and F4 side are talking to each other more, and all these sites are talking to each other more between exam 1 and exam 2, block 1 and block 2, block 1 and block 2. It looks pretty good. It looks like there's some pretty good brain training going on from the academic challenge of medical school. But we found something even more robust than that at F7 and F8. Now, F7 and F8 is the inferior frontal cortex compared to the other areas I just showed you, which were more prefrontal. And for the inferior frontal cortex, that seems to be associated with emotions, and emotional stability. More also syntax, understanding the meaning of words, and reading comprehension and understanding the meaning of terminology is very important in medical school. So it's a little bit different than what we saw before with the frontal. But look how robust this is. For theta, eyes open and eyes closed, the same pattern. For alpha, eyes open and eyes closed, same pattern. For beta, eyes open and eyes closed, the same pattern. Significant. So I think that's a pretty robust finding. And what we did next is we said, let's look at coherence overall. So we created a formula where we took the average of all the coherences at all the recording sites between the two hemispheres. And we said, how does that correlate with their Block 2 HSF exam score? Wow, very nice correlation, less than 0.05. Then we said, let's go to the next step. Let's do the same thing for intra-hemispheric. Let's take all the recording sites on one side and compare them to each other. And guess what? We also got a nice correlation between that and the exam score, telling us what? We think that, well, you know, if the different brain sites, either across the corpus callosum from one side to the other, are talking to each other, and within the hemisphere, more sites are talking to each other, recording sites, or the cortex underlying those recording sites are talking to each other. Maybe we're onto something in terms of the medical students' brains are getting trained to have more interconnectivity. And so when we looked at the beta coherence, so we didn't find any significant correlations with the exam scores, except for, as we said here, intrahemispheric. In terms of the individual site, the only one that was significant is the F3, F4 frontal or prefrontal site. So here what we see is, look at these people with the better coherence. They have the better exam score. You see they're kind of clustered here. And the ones with the poor coherence have lower exam scores. Again, it's very consistent. Just this was the only site that we were able to find this correlation for. So the next topic that we wanted to look at, or characteristic of the QEEG, is the asymmetry. So what does asymmetry tell us? It tells us about the voltage differences between two signals, between two recording sites. And we computed that. And I'm going to take the time as to create a disclaimer here. The disclaimer is, in the beginning, we really didn't know what we were doing too much, and we didn't read the literature as carefully as we should have. And so asymmetry, it turns out in the research literature, is calculated or computed based on the right minus the left. But because asymmetry, let's say alpha asymmetry, that's more left-sided, seems to be, and remember, the left hemisphere is the dominant hemisphere in right-handed individuals. In fact, even left-handed individuals, most of those have a left-sided dominant hemisphere. And so the left-sided alpha asymmetry, which is the one that's been most studied as a reflection of emotional state, seems to be associated with more approach behaviors, and the right-sided asymmetry, more avoidance behaviors, more positive emotion from the left side and more negative emotion from the right side in terms of alpha asymmetry. And so I said, okay, well, let's make the positive values for asymmetry left-sided, which means it was going to be F3 minus F4 or F7 minus F8. To me, that was logical. But I didn't read the literature carefully enough. And you see my note down here. We later changed the computation to be right minus left. And that's our learning curve, because when we read the literature, we find the people who are studying these kinds of things, that's what they do. So what I'm going to show you now is our calculation of left minus right. But it doesn't make any difference here. We can see from the brain maps that the asymmetry doesn't change much, does it? Theta, a little bit of beta asymmetry changing. So asymmetry, again, seems to be pretty much a robust trait of the brain that isn't amenable to too much change. So the overall change we think that we saw in the QEEG was more a reflection of a coherence and less as a change in asymmetry. So again, remember my calculation, left-sided, which this means that positive values, the positive values over here, see the cluster, are those people who have left-sided theta asymmetry. Okay, that's their dominant hemisphere, or theta is higher voltage. And when we looked at that versus the theta beta ratio, remember theta beta ratio was our indication that the higher the theta beta ratio, the poorer the attentional control perhaps, and the poorer their exam scores. So we see that the people who had more of this left-right frontal theta asymmetry, which means here's these negative numbers are indicating that the right is greater than the left. You see? So these are people with right-sided asymmetry. These are the people with the left-sided asymmetry that's associated with positive emotions and all of that. They had the lower theta beta ratio. Maybe better attentional control, and we know already better exam scores. And so we did the same thing here with the frontal theta asymmetry and the exam score. Here it's the theta beta ratio. Here it's the exam score. And we see a similar clustering and a similar pattern. So again, we just verified that the exam score was following the pattern we expect. Now, this is the most exciting part of the study, and I only have a little bit to talk about this, so that we will have some time for questions. So as we told you in the beginning, the experimental design was to have the subjects complete the individual self-inventory of psychosocial interactions from the New Mind Maps system, and do that within 12 hours after doing the QEEG. And so I made a table here that I took mostly from the New Mind Maps information. I reorganized it a little bit. Richard, I hope you don't mind. I might have reworded things a little bit. And again, I'm going to focus mostly on depression, on the depressed score. And remember, we took the graphical representation that you get in the ISI and we extrapolated it to numbers. Again, we hope that that was legitimate, and we think it was, but we'll see. And we can talk about it. So what we're interested in is the depressed score. Why? Well, you know, our counseling service tells us that we have a high percentage of medical students who are depressed, who come with needing counseling for depression. And that seems to be the big thing we're concerned about. And of course, occasionally in medical school, you have some suicides and you have some deaths and you have some all kinds of acting out and drugs, perhaps related to this depression. So we want to focus on that mostly. So remember, the ISI dimensions say that individuals who have negative thoughts about themselves, these are self-reports, according to the Beck's depression inventory, the correlation is greater than 0.8, according to New Mind. We want to focus on that. But again, you can read quickly through some of these others that I tried to make connect them. For example, anxiousness with depression and relaxed and inhibited, regulated, impulsive seem to be the kind of the opposites of each other. I tried to organize the table like that. And these are the other ones that you should be familiar with already. And this is again from the New Mind literature, showing their correlation coefficients with approach and avoidant behavioral paradigms. I got to get more information from you, Richard, someday about what these correlations, this is the reference population, a very large reference population, but what inventory they are against, they are correlated against. But we can talk about that later.
Mhm.
Well, this is our data, Richard. So this is what our data shows, that when we extrapolated the ISI data, okay, that we found correlations, all right? We found correlations. And these are the correlations. So for example, we're most interested in depressed. The correlation was between the ISI score and the F8, F7 frontal alpha asymmetry. Now, as I told you, alpha asymmetry is the asymmetry that's been mostly studied, reflecting positive emotions when it's left, negative emotions when it's right, approach behaviors when it's left, avoidant behaviors when it's right, watching TV shows or watching movies that are highly emotionally laden with emotional signals, negative signals from the right, asymmetry, and more positive signals from the left, and that kind of thing. So what do we see here? Now we changed the computation. So remember before we had left minus right, and now we learned to do it properly. So we've got right minus left, which means the left frontal asymmetries will have negative values when you see our correlation. And I'm not going to bore you, and we're running out of time anyway, with looking at the others, although we found significant correlations for F8 minus F7 with depressed, with relaxed, with regulated, with cooperative, and with dependent, and not with any of the others. And here's our data. So when we look at this alpha asymmetry, and this is only in the eyes closed condition. In the eyes open condition, we did not find this. And we looked at just that depressed score, because I told you, because of how much depression we see in medical school, we were most interested in that. So remember the negative values I told you are going to be more left alpha asymmetry, and look at these guys. These guys with the more left alpha asymmetry had the higher depressed scores. And the people with the lowest depressed scores had the smallest amount of this left alpha asymmetry. In fact, almost zero, close to zero asymmetry. So we think that tells us something about what we can expect in a larger population. We hope that frontal alpha asymmetry might be reflecting emotional state, as we saw here, and we could show you similar graphs for all of these, but the numbers will tell the story. So I'm finished, and so I want to acknowledge the American University of Antigua College of Medicine, which gave me the money to send to Roswell, Georgia for the equipment and the support we got from them, and my co-investigators, 4 medical students, and a junior faculty member. And so thanks for your attention, and we can open the floor to questions or comments, which I will appreciate.
All right,
Okay.
Rick, Richard's going to be unmuting people. That'll take us maybe a minute or so. One of the questions I had I was looking at your asymmetry data. One of the things we've seen with asymmetry, when you have a dominant pattern where it's on all the bandwidths, not just one or two, but like, delta, theta, alpha, beta, if it's all red or predominantly red on the left side, it's oftentimes an indicator of a brain injury with depression. and you mentioned depression with these students, which is interesting. On the right side, if it's predominantly red on the right in asymmetry, it's oftentimes indicative of a brain injury with anxiety. So I'm wondering if you would ask any questions about brain injury.
Yes, well, of course, yeah, there's a lot of data on that for brain injury. Remember, these are in our study. There's nobody brain injured here. They're all normal, and so showing that there's more left asymmetry. Now, what does asymmetry show? That's another important thing. We think from the studies, brain studies, that on animals and humans, that asymmetry at a particular site. Now you got to remember, we're looking at it at a particular site. Not looking at overall. So when we're looking at it at a particular site, like the F8, F7 I showed you, it tells us something about the specialization of that site. So that particular site, at least in our population, seems to be a little bit sensitive to negative self-reports, right? On the ISI, negative thoughts about themselves. So it's a little bit different. It's a little bit different than what you're talking about. Got to remember, this is site specific.
Okay, yeah. Well, just interesting to look at that as well.
Absolutely, absolutely, yeah.
I think other people should be unmuted by now, so we can let other people ask questions.
Yeah. Again, we did look at all those other sites, all those other frontal sites, mostly, and FP and F3 and F4. We didn't find anything. And probably if we look, and I don't remember, but I think we looked at the other sites, the other recording sites too, and we didn't find anything. We only found this F8, F7 jumped out. And what's interesting about F8, F7 jumping out, what's interesting about the theta-beta ratio jumping out at us, is that we weren't looking necessarily for it. We didn't make a hypothesis about it. We used a shotgun approach, and jumping out tells us there's probably something very significant there.
Well, Rick, this is really nicely done. I think it's great because it shows you how many dimensions begins to show you how many dimensions there are to an analysis like that, and you're just scratching the surface on your own data. It shows you how carefully you have to think through each thing and devise, what you're expecting to see, and then decide how you're going to measure it. And
how many mistakes you're going to make, like my computation with left minus right, to me was very logical, but I wasn't careful. I go to the literature. No, they do it the opposite way, right minus left. That was probably somebody like you, Richard, who's like a guru in the field, and they say, oh, we want to do it right minus left, and now everybody has to follow that.
Yeah, I mean, there's different ways to do it, and you have to make sure that you're comparing apples to apples and oranges to oranges, and there's still lots of controversy about all of these things, and you don't always find what you expect you're going to find. You think you're going to find one thing, and you don't. You find F7 and F8 instead, and it's like, well, what's that about? And then you have to think through, well, are there any exceptions? And Rob brought up, well, maybe there's a TBI thing there, but I think it's just great that you showed all these different ways of thinking about it, and how you use the statistics to ask each of these questions and answer them, and how much you have to do that just for, a small group of people. But it prepares you to say, what are we going to spend our time looking for when we get a lot of people?
Right, exactly.
And you got to do that first. And
exactly. And so now we're looking forward to purchasing a couple of your newfangled units that we heard great things about, and so we're going to move ahead with a larger population, honing in on some of these things with some fancier equipment.
That's great. That's great. That it's just so thrilling to see you doing doing all this stuff so carefully and so well, and thinking
Well,
it through and looking
Hi,
at
this
each
is
thing.
Dr. Steffens. How are you?
Yeah, please.
Hi, this is Jane Vanston
Yeah,
calling you back.
remembered your phone
Oh,
number.
I have to
The
turn
only thing I
somebody
forgot
off.
to ask you was the phone number for the insurance company
Okay.
itself.
Yeah, so the point is, yeah, I wish I was more careful. If I would have been more careful, I wouldn't have to change my calculation.
Yeah, well, it can be very confusing, you know.
It's a learning curve, and it's okay because when you publish it, you have to describe how you made the calculation anyway, so people
Mhm.
will be able to figure it out.
Yeah, yeah. Well, and again, that whole area is still fairly controversial. I mean, we're still working on how much do the asymmetries show anxiety and depression, and are there some areas more important than others? And still lots of work to be done in that, for sure.
Yes,
But
well, I'm very interested in the potential that we could use your ISI and some brain correlations kind of as a screening tool, you know?
I've already I published an article on that and showed how I arrived at that data, but it was different methodology than you did. But that can be helpful. But generally, you do find a pretty good correlation between anxiety and depression on the ISI and the EEG. But there are, from the data I've been running, you can see that there's lots of things that can confound it and confuse it. And I was just showing it on Monday night that we showed that people who are anxious tend to have higher anxiety score than people who are depressed. So depressed people tend to show lower anxiety scores. And once you get to a certain level of anxiety on the ISI over 25, those people start to disappear and show up as depressed people with anxiety. So when you're running samples in the 2, 3, 4,000 subject range. So you could and say, wow, there's a really complicated relationship between anxiety, depression, and that was going to confound some of the relationship between the ISI and the EEG,
Right. By the way, it will help us to not have to extrapolate those graphs if you can put numbers there.
Numbers on the actual graph itself.
Yeah.
Yes. That's probably a good idea. We could have sent you if you had asked us for those numbers, we could have sent you the numbers and how we calculated the dimensions. Always happy to give you that info, but we don't always generally stick it on there just because the clinicians we tend to eyeball it more than do the research numbers, but I'd be happy to send you those numbers. Okay. No problem. But yeah, please ask anytime you want to know.
By the way, as long as I got you on the line here, tell me about, remember I was bringing up, I didn't understand that that table that I took from you with the correlation coefficients against the against that reference population of a couple thousand people, which was very, very impressive. But I wasn't sure when you have the paradigm of approach and avoidance, what were they correlated with? What kind of inventory was the ISI compared to?
Oh, what that part was is we had a third party do that because we didn't want to bias it, and he took anxiety. I mean, he took approach and avoidance, and he went to see which of the positive and negative dimensions, like you mentioned, some of them they are all opposing. You've got approach and avoidance. You've got each dimension has two sides, passive and active in a sense. So he looked and he said, do the positive dimensions correlate with approach, and do the negative dimensions correlate with avoidance?
I get it now.
So we were trying to understand overall, do dimensions, social dimensions of behavior, positive and negative dimensions of behavior, do they correlate with approach and avoidance? So we were confirming that those were valid dimensions, because the idea was that in the future, you could use measures of approach and avoidance and compare that with the EEG and because that's how it originally was reported that way in the first literature, starting with Richard Davidson. So we were trying to establish, do social behaviors of approach and avoidance fall into those dimensions? And if they do, then we can use the dimensions of approach and avoidance, like Richard Davidson recommended, as the correlates of asymmetry. And that was the reason it was done that way.
Got it. So would the avoidant behaviors show a negative correlation where the
Yes.
approach behaviors would show a positive one?
Yes, that's why I built the ISI. It was built specifically to work with EEG, QEG research.
It's actually quite brilliant. Quite brilliant.
Well, hopefully it was done right, but we brought in some experts to make sure. And,
okay, well, we're going to keep validating it as much as we can.
Yeah, I'm just continuing Davidson's work and the others who followed, just following in their footsteps. And he's the brilliant one who came up with the idea,
So we hope to take this to a larger population and get rid of our type 2 statistical error and see if it holds up.
Yeah, it's very exciting. We're just thrilled
Run
to
it up
death
the
that you're doing
flagpole and see who salutes.
Well, you're right on the cutting edge of all this, Rick. Thank you so much for coming and sharing all those details with us.
My pleasure, my pleasure.
But we're out of time. We're over our limit.
Is Martin still with us?
Yes, I am, Rich.
Okay, good.
I have a lot of comments, but not here.
Okay, good.
Yeah, maybe we can pick up on this in the next open session. We can discuss more.
Yes, of course. And of course, my disclaimer is it's a pilot study. We really, don't know what all this means. This is all to develop novel new hypotheses to go further.
Yeah. Yep.
But Rich, quick appreciation. That's the way you went about it is very impressive. Congratulations for the systematic approach.
Yeah, thank you. Well, that's what we do in physiology. That's what physiology is all about. That's why I enjoy it.
Right, absolutely.
Yeah, but again, we're trying the best we can to see what associations we have that could be significant, because in human studies, we're never going to prove cause and effect.
Yes. Yeah, that's so true.
But associations are good enough for helping people. For helping people, helping clinicians develop the right paradigms.
Yes.
Train people.
Cause and effect is very difficult to show with human beings. It's so complex. We are not cannonballs that you can use calculus on hurtling through space.
Exactly.
Okay, everybody. We'll have to wrap this up because I know you all have to get back to your clients. Thank you so much, Rick. And maybe we can get you back to discuss some more of this sometime soon.
Sure, anytime.
Yep, thank you, Rick. Thanks, everybody. Bye-bye.